Um, JSU R. Watkins on YouTube. Or it's R. Watkins, it's R. Watkins JSU. But I'll post a link and look. Check your either check Blackboard or your gym accounts, and I will send out a link through Blackboard to you to look at that review. Okay, so kind of hit what we talked about last time with cell junctions, right? Cell junctions. Let me grab this light. Cell junctions are going to connect. Cell junctions are going to connect cells together. What we've been saying, cells are made of what? Cells combine to make what? Tissue, right? But they're not. They have to have some way to connect to each other, and that's where cell junctions come into place. 3 types of cell junctions we need to be aware of. The first is a tight junction. Key point here is it's a waterproof bond. It's a waterproof link. You're going to see tight junctions around your skin, for example. Okay, because you need to keep water. You need to keep things like water from coming in, but you also need to keep water from leaving you. So you have to have some type of waterproof barrier. That's where tight junctions come in. Another junction, adhering junctions. These are like spot welds. These are like points that are very strong that will not let cells separate. You're going to see adhering junctions in your heart tissue, for example. Because your heart's beating, multiple times a minute, 70 to 100 times a minute, depending on what shape you're in. And, those muscle cells are pulling apart constantly, right? There has to be some type of thing that holds those cells together to give them mechanical strength. That's what holds them together. Another one, gap junctions. These are like little holes between 2 cells. And we said that what? Tissue is when what? Cells get together and they start communicating and acting as one, right? Well, one way cells communicate with each other is through gap junctions. They can send chemical signals to one another, in some cases electrical signals, through these gap junctions, and they can choreograph their function. So the reason why tissue can be tissue is because the cells that make it up, they communicate, and one way they communicate is through gap junctions. So bottom line it, tight junctions, waterproof links. Adhering junctions, spot welds, like 2 bolts connecting cells together, mechanically very strong. Third, gap junctions. Gap, not where you get closed, but gap is in holes between the 2 where signals can pass between cells, where they can communicate with each other. Okay. Cytoskeleton. New topic, and we're almost done with this chapter, and we'll start the next chapter, and whatever we finish on the next chapter, that will be it for the test. Cytoskeleton. Okay, we have a network of protein inside of our cells. Now, I showed you that, that quote-unquote generic image of the cell before, and that's not really a true image because inside of our cells, we have this whole mesh network of lines and fibers that are connecting everything together. Don't worry about the second point. Let's just know what the first one is. That the cytoskeleton is protein filaments that are moving through our cell. Okay. So the first point is the important one. What is a cytoskeleton? That's what you should know. Now, let me talk about these points. Let me hit this point. One, I think the ones you should know is move organelles, move chromosomes. Those are 2 major functions you should know. We're going to hit those functions in later slides, but those are 2 major functions you should know. And again, we're going to hit that topic in a few minutes, so I'm just going to continue on, but you'll see those in the coming slides. Okay, so we have 3 types of cytoskeleton elements: microfilaments, intermediate filaments, and microtubules. Okay, so I want to talk about microfilaments. I'm going to talk about each of these, so you'll have a chance to write all of these down, okay? But I'm going to hit microfilaments first. So the first of that series is microfilaments. These are just thin protein strips made of something called actin. The role here is cell movement and mechanical support. When we start studying the muscle, actually next semester when we start studying muscle, you're going to see where this comes into play. When your muscle moves, it's going to have this actin as a part of your muscle movement. And we're not going to get into that today, but preview of coming attractions, you're going to see this when you talk about how muscles move. How your muscle moves uses actin protein. Okay, so we know it's cell movement and mechanical support. Okay, and what you should probably also know is microfilaments, and when you hear actin, think microfilament. Okay. So I want to show you how these microfilaments come together, and this is just to show you how it happens. So what's the protein that makes this up? Actin. So the actin makes this up, but how does that actin come together? How does that actin assemble? Well, how it assembles is actually it's going to assemble all by itself. It's going to self-polymerize. So all those globular particles are actin. What happens is that actin is going to come together, and it's going to form the microfilaments. So those purple and brown lines you see, those are microfilaments. And the little globular proteins coming together is actin, and that makes up the microfilaments. Okay. You can read that. Now let's not worry about that. You just know that intermediate filament. But I do want to talk to you about microtubules. Okay, these are made of a protein called tubulin. And these assemble and disassemble on the ends. So I'm going to show you how these things assemble and disassemble. So here is how they assemble. So all of those things are tubulin, and as they come together, they're forming a tube, which is a microtubule. That's the term microtubule. It is literally like a tube. Okay? Hollow on the inside, round tube. They depolymerize on the ends as well. So here's depolymerization. Watch what happens. Those little parts are tubulin, and now that microtubule is breaking apart. Okay. That's how it breaks apart. So what do we know? Microtubules are large. Of all those things we talked about, microtubules are the largest. Okay? They are made of tubulin, and they have multiple functions. The important ones here are separates DNA and mitosis and meiosis. That's important. That pathway for vesicle transport. I got a video for that. Pathway for organelle movement. There's a lot of functions that microtubules do. Has anyone, speaking of that separate DNA and mitosis and meiosis, has anyone ever heard of the drug Taxol? Breast cancer, yes. Okay, if you've ever known anyone with breast cancer, I believe Breast Cancer Awareness Month is coming up here. One common drug for breast cancer is Taxol. There's few other names for it. But how Taxol works is it interferes with the depolymerization of microtubules. So why is that important? So I guess the question is, what's cancer? If I say that, give me the thumbnail sketch of cancer, what is it? Other growth, exactly. Cells divide, and the checks and balances that stop cells from dividing are gone or inhibited. Therefore, cells are dividing all the time. We're going to learn later in the semester that DNA replication has to happen before a cell divides. So what if we could somehow block a pathway in DNA replication? That might block or prevent or inhibit cancer. Well, Taxol is going to prevent this depolymerization of the microtubules, and it's going to inhibit cell division, and therefore is a drug for the treatment of cancer, in particular breast cancer. Okay. So remember that last picture I showed you? It was like all flying apart, all that stuff. Taxol interferes with that process. Long story short, it's going to prevent the separation of DNA in mitosis and meiosis, and that's why it's used in cancer treatment. Okay, so let's talk about that third and fourth point, pathway for vesicle transport. Remember we talked about the vesicles last time? Those little sacks of things coming out of things. Remember that? Well, do those just like float around like balloons? Just willy-nilly floating around the cell? You think that would be a good mechanism for doing things? No, it's just they don't like float around like balloons and just randomly go where they want to. There is some type of plan, some type of organization for those vesicles, and that comes into play with the microtubules. Okay, so literally there are motor proteins attached to vesicles and organelles, well, vesicles here, and they will move those vesicles around the cell. Okay, so these microtubules are like a highway system in our cells. So let me show you. This big blue thing is a vesicle. Okay, that thing moving is a motor protein, and it's literally moving vesicles around the cell where it needs to go. Okay, it's not some random willy-nilly, I want to go where I want to go thing. No, vesicles start one place, and they're taken to another place along a microtubule network inside the cell. Okay. Another one, that pathway for organelle movement. Let's look at that one. So that's a mitochondria. Okay, and that mitochondria is also going to move around the cell. These things aren't like houses. Okay, they're actually moving objects around the cell, going where there's oxygen, going where they need to go. Okay, so again, the point we want to make here is that the cytoskeleton, in particular microtubules, are going to be a pathway, a conduit for moving things around the cell. Okay, finally, this is that part about the taxol, paclitaxel, whatever you want to call it. There's other names for it. But long story short, it's going to block the depolymerization of these microtubules, and it's used in the treatment of cancer, in particular breast cancer. Okay, so I'd like to, real world things. And if you've not had, no one has had my test, but I really like practical examples of things. I like biology things that are like, real world things, and to me that's real world things. Okay. So that wraps up that chapter, and then we're going to go as much through chapter 5 as we can get through. I'm not going to try to run through this so fast to make all of chapter 5. I doubt we're going to make it through all of chapter 5, but that's just the way it is. We'll only test what we cover in class. If we don't make all the way through chapter 5, we'll pick it up for the next test. Okay, I'm not going to artificially run through this lecture just to get it done. I want to make sure we understand it. Okay, new chapter. We're going to come back to the phospholipid bilayer. We kind of talked about the construction of the layer. Remember we talked about this when we talked about all the regions of the cell? We started on the outside. We said that's the layer. Then we talked about the nucleus. Then we talked about the mitochondria. And then we talked about the Golgi body and all that stuff. But we really didn't, we talked about the structure of the bilayer membrane, but we didn't talk about the function of the bilayer membrane. So that's what we're going to do today. Okay, we're going to talk about how this thing actually functions. Okay. Totally, this is just review. Don't write all this stuff down. It's just to review what we said before. Refresh your memories here. The memories are what we're going to refresh. The head's hydrophilic. It loves water. The tail is hydrophobic. It hates water. And that's why we have a bilayer membrane. I'm going to go past these because these are actually, I think, from your last lecture. I'm just throwing these in here just by way of review, just to set the stage. The important point here where we're going to start is the fluid mosaic model. You might as well star, underline, happy face those 3 words. How the membrane works is like a fluid mosaic model. This is how our membrane works. Now this, orientation is this. The blue is what? The blue is what? Hydrophilic. Those are the hydrophilic head. These red things are what? Hydrophobic. Are made of what? If I looked at those on a molecular level, what would these be? Lipids are, a little bit more specific, fatty acids. Perfect. Okay. So what do we see? Do we see this thing is like a piece of rock? No, it's more like a water bed, right? Well, that's how our membranes are. Our membranes are in this constant state of flux. Not only are our membranes always moving, but the proteins on the membrane have the capacity to move around. And we describe this ability to have motion as the fluid mosaic model. Okay. So all lipids and the proteins can move around the outer surface. Now I'm showing these molecules. I made this animation myself, actually. I fiddled with 3D animation. But anyhow, I'm showing these all connected, but in actuality, these molecules are moving amongst themselves. Has anyone ever been to Mardi Gras? Yeah? Okay, do you know what it's like as you get through the thing and everyone's like in the street moving around? You know what I'm talking about? And it's like you have to hold the person's hand that you're with just so you don't lose them in the crowd. If you think of that person as a phospholipid, that's how it's almost like, that things are just literally moving around. It's like that. Think of a huge crowd where it's like person on person, moving around. That's what the membrane's like. Okay? Okay, so, and to make the point, it's in motion, it's dynamic. It's not like a brick building, it's like a waterbed. Okay, so I'm just going to stop beating this dead horse. I think we get the idea. Our membranes are moving around. But here's the thing, motion is important for life. If it doesn't have motion, the cell dies. It has to have motion. Lots of reasons we're going to talk about. One reason is there's proteins on here, and for proteins to sometimes be functional, they're like floating around this membrane, right? And one's over there, one's over here. And they have got to come together, form a larger protein structure, and do a function, and then come apart. If they can't come together, no function happens. Nothing happens. The cell dies. It has to have this motion. Okay, so no motion, the cell dies. Okay, but the whole point of this, I know I said a lot of stuff, but the key point, fluid mosaic model, the membrane is in motion. Okay, so changes can happen to change the fluidity of the cell. One change is temperature. Okay, as temperature goes up, fluidity goes up. As temperature goes down, fluidity goes down. And I'm sure we've seen this. You take butter out of the refrigerator. It's in the refrigerator. It's what? Solid. You pull that butter out and you put it on the table, and what's going to happen? It warms up and becomes liquid. Bingo, there you go. Because the fluidity of those fatty acids has changed because of temperature. Okay. Now, let's use a more scientific example here. Cold weather fish versus warm weather fish. In cold weather, let me mention one other thing here. Cholesterol. Our cells have the ability to change our membrane fluidity, and they can introduce cholesterol. The more cholesterol, the less fluid our cells become. Okay, so more cholesterol, less fluid. So let's say if I take some cold water fish, outside of Alaska, it's in 40, 50 degree water, and then I compare that to tropical fish that are in 80 degree water. I would actually see a difference in the amount of cholesterol. The cold weather fish are going to be lower in cholesterol. The warm fish are going to be have more cholesterol. The reason is because the warm weather fish, their membranes are too fluid, so they make more cholesterol to decrease fluidity. And that's why warm weather fish are higher in cholesterol, because they have to make the cholesterol to maintain the correct membrane fluidity. I'm not testing on that part yet, but just an FYI type of thing. Okay. So let's use this little fictitious example and talk more about membrane fluidity. So let's say I have two cells, and by some miracle of science, I've put some markers on them. On one cell, I've got dark green markers, and the other cell, I got light green markers. We're good so far? Let's say by this miracle, I combine these two cells together. Okay, now what I told you about membrane fluidity, and these markers are moving around, what would happen when this makes one larger cell? Would it be half green and half dark green, half light green? what do you think it would look like? It would be a mix. And that's what happens. We see a mix because of the fluid mosaic model. Those spheres, we're going to use those, call those proteins here, those are moving around. And they're going to move around pretty much at random, and we're going to get a mix of those molecules. Okay. And again, don't write the bullet points down at the top. I'm just trying to show you what's happening here. As they come together, the fluid mosaic model says that molecular motions at random, and they're just going to form a mix, a hybrid of the two. Okay, membrane fluid functions require a balance in fluidity. Okay, there has to be a balance between strength and fluidity. You can have a situation where the membrane is too fluid. If that happens, then it's going to literally the thing will rip itself apart. It's moving around. Remember that picture of the blue and red dots moving around? Think of that to an extreme level. The membrane would literally rip itself apart if it's too fluid. Okay. On the other side, it has to have some fluidity. If it's not fluid enough, it's going to be like a solid shell. Okay. Both situations will kill the cell. Okay. So there has to be a balance in fluidity. Kind of review for the test. What did we talk about where we said that it was something like Goldilocks, where it was not too hot, not too soft, not too cold? Homeostasis, exactly. Same idea here. There has to be a balance between two things. It has to be just fluid enough. Too much fluidity, the cell rips apart. It starts getting fractures in the cell. Too little fluidity, and it's like a hard shell, and it does not function correctly. Okay. So basically what I said, not enough fluidity, that's bad. Proteins can't move around. Things can't come in and come out. So what do we know here? Not enough fluidity is bad. Cannot move proteins around. That's what you need to take home from that. Can't get things into and out of the cell. But on the other side, too little fluidity. Okay, yeah, I'm good. Okay, but we mentioned this before with the cholesterol. The cell has the ability to change that on a molecular level. Talked about adding cholesterol. If it's too much fluidity, the cell can add cholesterol, for example, and decrease the membrane fluidity. So the cell is not some participant here that just has to go with what it has. It can do changes to its membrane to increase or decrease the fluidity of the membrane. So it's not an inactive participant here. So what you should know, probably won't ask about cholesterol, but a question might be, can a cell take steps to change its membrane fluidity? Is that yes or no? Yes. Yeah, there are things cells can do to change the fluidity of its membrane. Okay. We talked about this before, and in that picture I didn't have these, but we have proteins inside of our membrane. Okay. Just wherever, reminder here, you see these little curly fry things. What do we call those curly fry things on protein? I can take a couple names here. We call those curly fry looking things on protein what? Let's pull out the Jeopardy music. There's a name we gave those things. Remember we talked about secondary structure? And we said that they were like sheets and helices. What do we call those? Those like curly fries. Begins with A. Well, they're made of amino acids, but we called those curly fry looking things a certain name. Alpha helices, remember alpha helices? Beta pleated sheets. Well, let's just call those secondary structure. Let's just go back to secondary structure. Remember how we said that a protein has regions where it can loop back on itself and form secondary structure? That's secondary structure right there. So this orange thing right here is secondary structure. As you get into biology, you're going to see that the secondary structures, particularly alpha helices, are really good for being what we call transmembrane proteins. They're going to go between the membranes. What you should know here, though, is proteins can fit between the cell membrane. It's going to become really important when we talk about transportation. It's going to become important when we talk about cell signaling. We're going to try to get to some of that today. But for now, we're going to say that proteins can fit between the cell membrane and they can move around. These membranes can move around. I kind of talked about that before, where membrane proteins over here, one's over here, and they can come together and move around. Okay. Wow, there's a lot of words there. Let's not learn all of those. Let's not learn that whole that table is just for review. Okay. Let me skip past to where I want to really start talking about. Okay. Before I do that, let me just hit this point I talked about before. Remember we said that the membrane is like the face of the cell, right? We talked about this I believe a week ago. You know, we have a mouth. We can let things in and let things out. Well, the proteins on the cell are kind of like that. We call them channels or pumps. They're going to be a conduit for letting things into and out of the cell. We have nose and ears, eyes. We can detect things. The thing with the cell is it has receptors on the outside where it can detect compounds outside and tell the inside of the cell what's outside. Good example of that, you guys heard of the hormone insulin? Okay. Insulin is going to fit outside your cell. There's going to be a receptor outside your cell, and then it's going to go through the cell membrane, and it's going to make a change inside your cell. And the result of that change will direct that cell to do biochemical processes that will control your blood sugar. The shape lets others recognize you. There's something called MHC markers on your cell membrane that identify that cell and the tissue that's made up of those cells as cell tissue. Does anyone in here know anyone that's had a transplant before? Organ transplant? Well, they have to take what, usually? What kind of drug? Immunosuppressive drugs. Because when they get an organ, they're going to get something that's not shaped like their body, like their MHC markers. And they have to tamp down or inhibit their immune system to prevent rejection, because that last point, the shape, it's a different shape on the outside of the cell, or on the outside of the cells that make up the organ. And because of that, your immune system can actually attack that organ. So that's why you have to take immunosuppressive drugs. So let's talk about each of these somewhat in a little bit of detail. I'll skip this one. This is the one I want to talk about. All of these are important, but I'm just going to talk about some of the more important ones. One thing that can be on the outside of a membrane is a receptor. And this can pick up signals from the outside and tell the inside, hey, something's happening. So for example, we don't see one up here, but let's just say a hormone, here's a receptor. A hormone on the outside fits into the middle of this receptor. This would then go through the cell, and on the inside, cause a change inside the cell. It would elicit some type of a response. Good example again is insulin. Okay, insulin does not enter into your cell. It stays outside. It's what's called a peptide hormone. Too big to get inside the cell. It has to sit outside the cell and bind to a receptor in order to work. Another important membrane here is a recognition protein, like MHC markers. These tell your body this cell is a self cell. Don't attack it. I didn't mention MHC. We might mention MHC later on, but for now we're just going to say that how a cell recognizes what it is by recognition proteins. Okay. And for this, just know that this is how it identifies it as a self cell. That's the key point here. Another thing we see is a transporter, a channel. a tunnel. Sometimes we've got to get stuff from the outside in, and sometimes we've got to get stuff from the inside out. But that stuff can be too big to pass through the membrane, so we have to have a tunnel in a sense, a pathway to go between the outside to the inside. And that one of those we're going to see today is a passive transporter. And we'll fill in what that means later, but let's just say right now that's just a tunnel between the two sides of the membrane. Now that high-low concentration, we're going to talk about that later today, but for now let's say that a passive transporter is like a tunnel, a pathway for molecules to move from one side to the other side. Another one is an active transporter. Same thing, it's also a tunnel, but the direction of movement is different. And I know that's kind of a vague explanation, but we got a lot more slides on this later. But for the here and now, let's just know that there's different types of tunnels between the membrane. Okay. Now, those last 2 points, we're going to start talking about those a little bit more. It's transport across a membrane. How do we get stuff across a membrane? Because you got to think how a cell works, right? It has to get stuff into and out of a cell, right? Well, that's what we're going to talk about. How does it do that? Okay, so how do we get stuff into and out of a cell? And to talk about that, we've got to do a quick review on concentration and concentration gradients. So this is where we start writing, right here. What is a concentration? Concentration is not when we think about something a lot. That is concentration, but not what we're talking about here. Concentration in this class is the number of molecules per unit volume. So the diagram up here, let's say that yellow is ions or molecules, and we have 3 containers, right? Something that's high concentration has more of those particles per the same area than something that's low concentration. Now this is just a nice little example to introduce you to this. Then we're going to take this and apply it to a membrane. Okay? So now let's make a little experiment here. Let's take that high concentration container on the far left and link it up to that low concentration on the far right. What's that going to look like? So we could somehow link these two together. Right? We've got a container with high concentration on the left side, low concentration on the left side. I'm sorry, the right side. High on the left, low on the right. Now let's say if this pathway between them, there is some type of a spigot, some type of a valve, and we opened up this valve right here. What's going to happen to the concentrations in each of these? One's going to become less, one's going to become more. So these yellow particles are going to move around in such a way until what? They're going to equalize out. We call that equilibrium. And so the particles are going to move from high concentration to areas of low concentration. Now in science, we have a name for this. You might write this down too. You want to write this down. We call this going down the concentration gradient. Continue in biology, you're going to hear this term thrown out a lot. People are going to say, does it go down the concentration gradient? Well, this is going down the concentration gradient, where things go from areas of high concentration to areas of low concentration. And that's down the concentration gradient. And obviously what's going to happen, they're going to stop, and everyone has said this before, they're going to stop when the concentrations are the same. Now, technically, there can be movement. I'm not going to test this. This is just FYI. Technically, you can have movement between the sides, but the net change in concentration stays the same. So I can't say that there will be no movement. But for every movement from left to right, there'll be one movement from right to left. But we're going to say that they're going to equal out. Okay, so a concentration gradient, write this one down, this is a difference between the two sides. Okay, so if we have a concentration gradient, if I say to you in a biology class, there's a concentration gradient of these ions on the membrane. What I'm really saying is we have two different regions of concentration. One region has a very high concentration, the other region has a lower concentration. So if you hear the term concentration gradient, that's all it is. Okay? One has a lot of something, the other has very little of something. Now, let's say I open this spigot up right here. Stuff goes through. What would we call that? So if I opened up the spigot here and stuff comes through, what would we call that movement? We call it going down the concentration gradient. Exactly. Exactly. Okay, so that's the concentration gradient. And over time, we see what's going to happen. Molecules are going to go down the concentration gradient until there's no concentration gradient at all. Okay, when there's no concentration gradient, we've got equilibrium, no net movement. Okay. Now we got a special name for going down the concentration gradient. We call that diffusion. Diffusion is the net movement of molecules or atoms down a concentration gradient. So that process that we know what will happen. If I open this spigot up here, the stuff goes down the concentration gradient. What's the process by which that happens? Diffusion, exactly. So that's diffusion. So someone says diffusion to you, you know that what's happening. You got two areas, one's more concentrated, one's less concentrated, and just by stuff that you already know, high concentration is going to go to low concentration down the concentration gradient, and that whole process right there is diffusion. So that was before diffusion. After diffusion, it went down the concentration gradient. All right. Okay. So why is this important? Who cares? Right? Well, we have to know. That's a good question. Who the heck cares? Well, we've just had these little containers of things. But instead of having containers of things, what if this container was outside the cell? This container is actually inside of the cell, and this area here is the bilayer membrane. Understanding how this works helps us understand how ions and molecules move through our bilayer membranes. Okay, so now just kind of retrain your brain. Inside the cell, outside the cell, bilayer membrane. The same principles are going to apply. Okay. So the direction a molecule moves across the cell depends on the concentration gradient. Does it go inside the cell? Does it go outside the cell? All of that has to do with concentration gradients and the differences of concentrations, be it atoms, molecules, small sugars like glucose, things like that. Okay. So bottom line is, why is it important? It tells us the direction a molecule will move across the membrane. Okay, so let's talk about movement across the membrane here. Okay, we got 15 minutes. If we don't finish all these, we will only test what we finished today. Okay, so we've got 4 ways. Ignore that 3 ways. It's 4 ways. We've got 4 ways molecules can enter into a cell and exit from a cell. 4 ways. And I'm going to talk about each one of these ways. So I'm going to go to the next slide, but it's the same one. And I'm going to return to this slide over and over. So don't worry about writing everything down. You're going to get a chance to write this down over the next 10 minutes. So the first one we're going to talk about is going directly through the bilayer membrane. So why don't you write down right now, instead of writing all these down, just write down, he's talking about going directly through the bilayer membrane. Okay, so let's talk about going directly through the bilayer membrane. This is just the diffusion we covered before. This would be one of those gray chambers, and this would be one of those gray chambers. That empty zone between them is the cell membrane. So if there's a lot of yellow dots in the blue area and a few yellow dots in this pink area, what's going to happen? What direction is it going to move? What would we call this? down the concentration gradient, or another word, begins with D, diffusion. Exactly, so that's diffusion. That's one way things can move through cell membrane. Okay. So again, we're moving substances down their concentration gradient. The process by which we do that is diffusion. Okay. So this is before diffusion. What's going to happen after diffusion? What are those yellow dots? Which one? What's going to happen to those? We have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. We have 14 yellow dots. How many yellow dots will be on the top? 7. And how many on the bottom? 7. There we go. Just like magic. Okay? That's going down their concentration gradient until they have reached equilibrium. Okay? So they're literally scooching between the fatty acid tails. They're not going through any kind of tunnel or anything. They're just literally just pushing their way through those fatty acids. Wow, that's a lot of writing there. Let's not write all that down. Let me just hit some high points. The idea here is what? Let's go back to this picture. What about those yellow dots allows them to do diffusion? Okay. What about those yellow dots? Size. That's one thing. The bigger they are, think about the membrane. These little phospholipids. If I got something huge, it's going to have a hard time scooching its way through that membrane, right? So the larger it is, the slower it happens. Smaller it is, the faster it happens. Another important one here is the steepness of the concentration gradient. If I've got a massive difference, one is 10,000 times more concentrated than the other one, just because it's so much more concentrated, it will go faster down the concentration gradient. Now, if there's a little bit of difference, it's going to go slower. So that's what I mean by steepness of the concentration gradient. If I have super duper high concentration here, low concentration here, it's going to go faster. If I have concentration that's maybe 10% higher here on the outside of the membrane than on the inside, it's going to move slower. So that's what I mean steepness of the concentration gradient. Then the other one I want to hit is charge. That's another important one. Molecules that are not charged, like gas, oxygen, carbon dioxide, those have a very easy way of getting through. Hormones, lipid hormones, testosterone, estrogen, things like that, those are nonpolar. Those get through relatively easily. Charged molecules such as sodium, potassium, if they have some type of charge on them, they're just going to have a really rough time getting through. To the point where they can't really just go through by simple diffusion through the membrane. They have to have something else. So the three ones you should know here is size. I think that one makes sense. Steepness of the concentration gradient, and charge. Okay. So some materials pass through the bilayer membrane. We just said that, the uncharged ones. But some do not pass through that bilayer membrane. Okay, so point here is some things do pass through, some things do not pass through. And call this whole, this control selective permeability, where there's some control of what can move through the membrane. Okay, so you might want to say in your notes, selective permeability, some things can move through, some things cannot move through. That's basically what I want you to know. Okay, so I guess the more important thing here, and you should know this, is what does and what does not move across. Well, what does move across are gases, very small nonpolar molecules, and water, to a degree. But let's just say gases like oxygen, carbon dioxide, nonpolar molecules. Some things have an easy way of getting through this membrane. So for this, what moves across it? Gases and nonpolar molecules. That's what can easily do this diffusion. But what cannot? What does not are ions. Those are charged molecules. Calcium is an ion. Potassium is an ion. and polar molecules, things that have a charge on them. Okay, so things that go through, nonpolar and small molecules, things that do not, ions and large polar molecules. Okay, so this is our second point. I think this will probably wrap us up for the test. We've got 7 minutes here, and I'm not going to run through this too fast. So we've said that some things cannot just go directly through the bilayer membrane, right? Well, there has to be an alternative way. And we talked about channels. Remember I said there's like tubes in the membrane? Well, another way these things can go through are through tubes. We call these channels. And we call this process, and you might want to write this down under here, facilitated diffusion. So if I facilitate you, what am I doing? If I'm a facilitator, what do I do? I'm helping you out, aren't I? I'm helping you out. Well, the facilitated diffusion is going to use channels to help these big polar guys out, help these ions out, do diffusion. Now, the stuff we talked about diffusion, going from high to low concentration, down the concentration gradient, still applies. But instead of trying to scooch its way through the cell membrane, there's a tunnel, a hole, a little channel like a straw that it can go through the membrane. Same direction, slightly different mechanism. So this is how it works. On the left, the red dots, that's simple diffusion. On the right, the green dots is what? Facilitated diffusion. How are these similar? What's the similarity here? How are they similar? Exactly. Concentration gradient, high to low, high to low. How are they different? Exactly. One's going through a tunnel, facilitated diffusion. One is trying to scooch its way through the bilayer membrane. Simple diffusion. What kind of molecule would this red guy be? Gas or something else? Nonpolar molecule. What kind of molecule would this one be? Ions or large polar molecules. Glucose, potassium, chlorine. Just a quick clinical note here. Cystic fibrosis. There's a chlorine transporter like that in cystic fibrosis, and people that have CF actually have a malformed channel like that. So, FYI. Okay. So the point I'm trying to make here is this is facilitated diffusion. It goes down the concentration gradient, but it goes through a tunnel. It's being facilitated. It's being assisted to go through the membrane. Okay? We got time for one more, and this is a real fast one. Maybe even one more for that, but let me just hit this one. I got to hit this one. Sometimes we got proteins, huge proteins, that are just too big to get in and out through those tunnels. So we've got to do something else. And that is vesicle transport. We're going to put them in a small little ball and either bring them in or take them out. We call those balls vesicles. What can you see here? Bacteria, large bacteria getting into the cell. Incredibly important in the immune system. Peptide hormones. If there's a hormone that the cell is making and needs to release, it needs to put in one of these little balls to release that hormone. Okay, so lots of times we got to put them in these little packets, these little vesicles, these little balloon-like shaped objects to put them out or bring them in. Okay, and that whole thing is called vesicle transport. Okay, this will be the last 2 points I want to make here. I got a little video to make the point, and then that's the test. You should know what endocytosis and exocytosis is. But you know what? This is pretty easy. Endo means what? Inside. What's exo? Outside. So going inside to the outside, 1, 2, 3 is exocytosis. This is inside the cell. The blue is outside the cell. Exocytosis is have a vesicle, and it fuses with the outside, and it releases stuff to the outside. It's exocytosis. What do you think endocytosis is going to look like? Opposite, exactly. Wow, that's creative. Let's flip that picture. Make 1 and 2. 1, 2, 3. Large molecules on the outside. brought into a vesicle and then brought into the cell. That's endocytosis. You can put your pencils down. Got a movie here, and then you guys will be out of here. So this is endocytosis. So we've got a bacterium here. This is what it is. Blue is outside, pink is inside. What happens? It has to form a little vesicle, a little ball to bring that in. That is what? Endocytosis. We're bringing stuff into the cell. Okay. Yeah. That's exocytosis. What's exo going to do? Take it out. It's going to go from the inside out. These things are too big to go through channels. It has to go inside of a membrane ball. Okay, and then look for the review probably later tonight or probably sometime Saturday. I will check your gym email. I will send out an email the second I upload it.